Hello everyone, I am Ruchi Gupta, working as an Assistant Professor, Department of Pharmacology in Smriti College of Pharmaceutical Education, Indore. In today's session, we are going to discuss about the start from glycoside. Glycoside are the organic compounds present in a lot of plants, some animals. These compounds, upon hydrolysis, give one or more sugar moiety, is also known as glycol part, from and non-sugar moiety is also known as aglycol and gelatin. Okay, that means organic compound on hydrolysis gives sugar and non-sugar moiety. Okay, non-sugar moiety. Sugar moiety, it is responsible for the adsorption, distribution of the gelatin part or aglycol part. And aglycol part, that means non-sugar moiety, is responsible for the therapeutic action and pharmacological action. Then sugar and non-sugar moiety both are made by oxygen linkage. Then next, physicochemical property of glycoside. Glycoside are the colorless and non-volatile in nature. But flavonoids is found in yellow in color and anthocyanin is red or orange in color. And it is solid amorphous in nature. It gives positive reaction with Mollis test or Mollis reagent and Ferrin solution test upon hydrolysis. And they are soluble in water and insoluble in organic solvent. Most of glycoside bitter in taste except glycyrrhizin and stigosides. Both are sweet in taste. It is odorless except saponin. Then function of glycosides. It is converting toxic material to less or non-toxic material. And it is a source of energy. Then it is also used for storing harmful products such as phenol, and it is also used for the regulation of certain function, that means it is used for the maintenance of the growth and regulation of growth. And some glycosides have beautiful color, so it is used for the pollination process, and some glycosides have antibacterial activity, so they protect from plants, from bacteria and some diseases. Then classification of glycoside on the basis of linkage between the glycol or aglycol part. So first category, C-glycoside, in which glycol is linked directly carbon of aglycol part, and it is also known as allyl glycosides. They do not hydrolyze by acid or alkali. Then examples of C-glycoside, allyl is the main active constituent in aloe vera. So allyl is a type of C-glycoside, and cascaroside in cascara. Both are examples of C-glycosides. Then next, S-glycoside, that means sulfur glycoside. Sulfur glycosides or S linkage is mainly found in isothiocyanate group or glycosides, in which glycol is linked with sulfur of aglycol part. And example of S-glycoside, cynigerin from black mustard. Then next, N-glycoside, in which glycol is directly linked with N part of aglycol, and nucleoside is the example of N-glycoside linkage. And nucleoside include DNA and RNA. Then next, O-glycoside. O-glycoside means glycol part, and they are hydrolyzed with water, with alkali and acid, and sinusoid is present in the senna and renin from rhubarb. Both are example of O-glyco. The next classification based on chemical nature of A-glycon of glycosides. Then first, anthraquinone glycoside. Here A-glycon contain anthraquinone and its derivative, that is anthone, anthranone, dianthone and dianthone. It is mainly found in dicot plant except Liliaceae family. They have laxative effect. Then examples of anthraquinone glycoside: sinusoid from senna, aloin from aloe. Aloe vera and senna both are example of anthraquinone glycosides. Then next category, esterol or cardiac glycoside. Here A-glycon contain steroidal moiety, in which C13 or C14 containing steroids because of either 5 or 6 member lactone ring respectively. Cardiac glycoside with 5 member ring, lactone ring, is called cardinolides. Glycoside with 6 member lactone ring is called bufotenolides, in which the sugar part is attached to C3 carbon by beta linkage, and different type of sugar like glucose, fructose, rhamnose, GD-toxose, pivotose are present in cardiac glycosides. They have cardiac action, so it is used in the treatment of congestive heart failure and cardiac arrhythmia. Purpurea glycoside from Digitalis and Thivantine from Thivetia. So Thivetia and Digitalis both are example of cardiac glycosides. And then next, saponin glycoside. Saponin is Latin, means soap like. Okay, here A-glycon contains sapogenin. Sapogenin is a high molecular weight substances which by acylation gives crystal forms. They are hemotoxic in nature, so they are which is lysis of erythrocytes. The harmful saponin is called as sapotoxin. According to the nature of A-glycon, saponin are categorized into two groups. First, steroidal saponin. It is also known as tetracyclic tritropanoid saponin. Then second one, tritropanoid saponin, also known as pentacyclic tritropanoid saponin. So, steroidal saponin, it contains sapogenin with cyclopentophenanthrene nucleus. It is linked with sugar and it is distributed in Leguminosae, Solanaceae, Apocynaceae family. It is used for the synthesis of vitamin D, sex hormone, cardiac glycoside and corticoids. Then examples of steroidal saponin: diogenin from Diosporium and hecogenin from Safed Musal. Then second category, tritropanoid saponin. Pentacyclic tritropanoid saponin contains sapogenin with pentacyclic tritropanoid nucleus. It is linked with sugar or uronic acid. It is found in Polygonaceae family, Barbidiaceae family and Umbelliferaceae family. It is used as a anti-inflammatory drug, or it is used as an anti-inflammatory. Glycyrrhizide from liquoriza and acetylglycosides from brahmi. So liquoriza and brahmi both are the example of tripropionate sap. Alcoholic glycoside. Salicin is an alcoholic beta glycoside found in the genus of Salix. Salicin is in the body is converted into salicylic acid, which is closely related to aspirin. So it is used as an analgesic, antibiotic and anti-inflammatory activity. The next, phenolic glycoside. Here a glycol contains phenolic group and berberine or Castophilus uva or C contain albumin. It is used in a urinary antiseptic effect. The next, coumarin and furanocoumarin glycoside. Here a glycol contains coumarin. It is exists in free form or in glycosidic form. Furanocoumarin are formed by fusion of furanate to coumarin at 6 and 7 and 7 to 8 position. Then tocophenes contain coumarin. The next, isothiocyanate glycosinolate glycoside. It's a glycol part contain isothiocyanates. It's as the name implies, sulfur along with nitrogen. Cinnamon is found in a black mustard. Then next, flavanone glycoside. Here a glycol contains flavanone and it is occur either in free state or as a glycoside. It contains 15 carbon skeleton which is consist of 2 phenyl ring, that is A and B, and a heterocyclic ring, a 3 carbon rich carbon. So the carbon structure can be abbreviated by C6, C3, C6. Then most of flavanoids have carbonyl functional situated at one end of the bridge, and they have antioxidant effect. Rutin from buckwheat and quercetin from ginkgo. Rutin and quercetin both are the flavonoglycosides. Then next, iridoid glycoside contain aglycol iridoids. Iridoids are the type of monoterpenoids in the form of cyclopentanocarbon. Most often as glycoside iridoid, aglycol linked with glucose or sugar. So, octone and catechol. And catechol both are the example of iridoid glycoside. Drug powder undergoes the extraction process by using alcohol solvent. Due to high heating, deactivation of enzyme in the drug may occur. So, maintain the temperature below 45 degrees centigrade. Then it is filtered. Then filtrate added lead acid in which precipitate the tannins and eliminate non-glycosidic impurities. The excess of lead acetate is precipitated as lead sulfide by passing through hydrogen sulfide gas through the solution. The extract is filtered and concentrated to obtain crude glycosides. The crude, then isolation of glycoside. Different type of active constituent isolated from the crude extract of glycoside by fractional solubility, fractional crystallization and and chromatographic methods, in which TLC and column chromatography mainly preferred. Then identification test for glycoside, Braun-Tager test, modified Braun-Tager test are used for the identification of anthraquinone glycosides. Killer-Klein test, Legal test and Valger test are used for the identification of cardiac glycosides. Sodium picrite, mercuric acetate test are used for the identification of cyanogenate glycoside. Froth and pneumolization test is used for the identification of saccharine glycosides. Alkali test and fluorescence test are used for the identification of coumarin glycosides. Synodon, alkali and lead acetate test are used for the identification of flavonoid glycosides. Glycoside is mainly used as an anti-inflammatory and analgesic action, and it is used as anti-rheumatic and purgative and cathartic, cardiotonic, anti-ulcer, expectorant, antidiuretic, and it is also used stomach ache. Thank you.